Hi guys, I promised you a test take up. I finally got my phone unloaded and ready to go, so here it is. The knowledge and understanding section, you should be able to see the correct answers there, so I'm not going to talk about that. Thinking and inquiry section, again, I told you it was going to be lab-based, and it was. The potato question was about a sweet potato, so you should have been going, okay, simple sugars for the sweetness, and potatoes are full of starch, so I asked specifically for the types of carbohydrates there. So some people told me about proteins and lipids, didn't ask for it. So simple sugars, obviously the Benedict solution, and then for the starch, the Lugol's iodine, and you had to tell me the positive and negative results in order to get full marks for that one. The next question was salad dressing question, and the salad dressing is hypertonic. Oil, sugar, vinegar, anything that's not water is going to lower the concentration of water in that dressing, and so what's going to happen is the water inside the plant cells is going to leave and go out towards the dressing, which will indeed dilute the dressing slightly, but more importantly, it will plasmolyze the cells. They'll lose all the trigger pressure inside, and there won't be that support of the pushing out on the cell wall, and then you get the gross wilty salad. Then we had the blood question. If you were to intravenously, so introduce right into the bloodstream some distilled water, we would have a problem because, of course, that would be hypotonic, and so blood cells would actually take on so much water that they would rupture. There's no cell wall to stop them from expanding, and then you'd have cell lysis, which is certainly a dangerous condition. A lot of you said, hey, a saline solution would be great, and yes, it is called a saline solution because it's got a little bit of salt in it and a tiny bit of sugar in there as well, glucose, but what we're looking for there is that it is isotonic. So those saline solutions are set up such that they are isotonic or slightly hypotonic to the natural blood composition. Communication questions, you did a great job on your diagrams, not really worried about that. We had the enzyme question, so I was looking for specific vocabulary there. You need to tell me that enzyme B had the lowest activation energy, and then I asked you what type of reaction. It was a hydrolysis reaction, and if you look at the formulas, you should have been able to recognize that it's a disaccharide breaking into a monosaccharide. Your compare and contrast question, you have to be specific, please, with your similarities, as in, I don't want to hear things like, oh, both are needed in the body. Not specific enough, okay? Give me something a little bit more concrete than that. And then also with your differences, make sure you're giving me contrasting sets of points. So if you say one thing about DNA, tell me something about protein that would match up with that. And that's how you get a full mark for each of those. But those questions are really well done anyhow. Finally, we have the the making connections section. And we had the question about amphoteric molecules, just like phospholipids. I'm talking about soap and lecithin. Those two things are not forming membranes like we have in living things. Some people got a little confused with that concept. What they are able to do, though, is grab something fat and something water and pull them together, right? The hydrophilic and the hydrophobic portions, like attracting like. We had the egg white question, so you need to tell me specifically that if we put it in vinegar, we're not talking about the shell here, if we put the egg white in vinegar, it would go hard and it would go white and opaque, just like as if you had cooked it, because the same thing is happening. Heat and vinegar, pH, low pH, will denature those proteins. Why doesn't it go back when you cool it off? Because that is a permanent physical change. What you've done is you've actually broken and rearranged bonds, and so your secondary and tertiary structure are altered. So that's it. It's done. You can't change it back. Finally, we had the nerve gas question. Now, this was all about competitive inhibition. You had to tell me about the active site and the fact that that nerve gas is going to go in and take the spot that that neurotransmitter acetylcholine normally takes. So that enzyme now is not breaking it down like it should be broken down, which means that neurotransmitter just keeps working and working and working and working, and you have overstimulation of the nerves, and that's what causes the problem with that. That last little piece was a little bit hard to get, but you should have easily recognized the hints I gave you about competitive inhibition. Any questions about the test, please don't hesitate to let me know. Please, people, take the time to read the feedback. You never know when you're going to see these questions again.